Okay, so if we're here at the beginning, you guys are going to want this lab open and on your screen, and you guys are going to be working together in breakout rooms on question 3. See if you can figure out if this amount of caffeine is having an effect on cell division rates. And again, to kind of help you with that, let's have this pre-lab handy so that you guys can look at what we did last class, because you're going to do something similar. So again, to kind of review what we did here is I've given you basically 2 sets of numbers. I'm providing you with normal cells. And we have a certain number, I counted them up, of these normal cells only exposed to water. Put your phones away for me, please. 31 of them were in interphase and 9 of them were in a stage of mitosis. And I was able to figure out the percents of the cells that should be in these 2 stages if they're dividing normally, because these were the normal cells. I've given you numbers of cells that have been exposed to a chemical. Okay, so I'm using the numbers from the normal, the percents from the normal, and the numbers from the chemically treated cells to fill in these first 2 columns. Okay, when you get into your breakout rooms, I want you guys to see if you can at least figure out the numbers that are going to go in those 2 columns so you can make a decision about this caffeine and if it's affecting cell division rates. Let me post and open up in a second those breakout rooms. I do not want more than 5 people in a breakout room. Why? Because sometimes I have like 14 of you in one room and there's only like 2 of you figuring things out and the rest of you are just along for the free ride. Okay, so you can use hand signals to let people know which room you're going into, and I want no more than 5 in a room. Also, before we go to our rooms, some important announcements. I'm going to postpone your test to Tuesday of next week. So that means that Unit 5 test is going to be the first grade to go in the grade book for semester 2. I asked third block about it, and that's kind of what they went with. They're like, why don't we just let it be the first grade in semester 2, okay? So that test is going to be taken in class on Tuesday. So the first day I have you guys are coming into a test. Which one, I think that's going to give us more time to study, because I wasn't going to give you guys a day of review, and I think you do need a day of review because we're just not able to go as fast as what I always want to go. And second, I am much more happier about you guys testing in person, because I can tell without a shadow of a doubt that I have some people that are cheating with different screens pulled up. And it's on a different device because it's not being detected by GoGuardian. And I'm currently working with a tech specialist because I'm like, it's very clear that person was cheating. I was watching her on Meet the whole time, so she was looking at her screen. And also, I was running GoGuardian, so I could tell she didn't open any other tabs on that computer. So I have no trust whatsoever, and I'm glad that we're going to be testing in person. Okay, so just note that's going to happen on Tuesday. All right, guys, let's go ahead and get you in your breakout rooms, and I want you guys to be trying to solve question number 3, trying to figure out what the first columns of that chi-square is supposed to look like. We'll see you there. All right, just waiting for everyone to get back on here again. and let's just kind of take a look at how you guys did. Okay, so hopefully you kind of ended up with something that looked like this, right? From the control cells, you figured out these percents. That normally 86% of those cells were in a stage of interphase, and 14% of those cells were in a stage of mitosis, okay? And then these numbers were the 1 micromole caffeine treated numbers, okay? Is that right? Does that look right to y'all? I feel like my numbers are off. Do you have to, okay, just making sure, all right? So then total, I'm just gonna add them up. This is how many cells, oops, that's a 4. 341 cells were treated with caffeine, that amount of caffeine. So again, to figure out these expected numbers, you have to take your percent times your total, and that'll give you that number. Percent times your total, and I'll give you that number. Okay? Did anyone have those two numbers?
Hold on, give me a second.
You're there, Aiden.
For me, I got, for interphase was 293, and for mitosis 448 or 40. Yeah, 48.
48?
Yeah. Make sure that's right.
They add up. Okay. All right. So I'm just curious. Let's just look. This is how many cells were in interphase and mitosis when they were treated with caffeine. If they're dividing normally, this is what we'd expect. Do you think those are a good fit? Do you think these number sets are close enough to each other? Okay. So again, that's what our chi-square is going to help us do, is make this decision the same way. All of us are going to make the same division or same decision. I'm going to guess, did anyone actually work this out? I'm going to guess you did.
Yeah.
Was it a poor fit then?
Yeah.
Man, it's really close. I think this is a one that's, I mean, you could go either way with it. Okay, so you determined it was a poor fit, which means that if these caffeine numbers are not fitting the normal division numbers, that means they're not dividing normally, and the caffeine is affecting the division, okay? All right, let's get rid of that, and let's go into this next lecture, which is 5.3. So you might want to have 5.3 handy. And let me do a second just to kind of close up some of my extra screens here that are lagging me a little bit. And you need to grab 5.3. Okay. So I think the best way to teach replication is to watch videos to get a sense of what's going on. But I'm going to use one diagram here to start with probably the most basic and important information that you have to figure out. Because if you get kind of this first step wrong, all of the questions that you're going to answer are going to be counted wrong because you have not figured out which is the leading and which is the lagging strand. Okay. All right. Where are y'all at? There you are. Make sure I'm recording. And it looks like I am. Okay. So here's the thing is I'm going to ask you which side of the DNA that's being replicated, A or B, which one's the leading strand, which is the lagging strand. And before you can figure that out, you have to know something kind of important about DNA. We've mentioned this before, is that if this is a strand of nucleotides, the ends, when you get to the end, the nucleotides that are at the ends are going to look different, just like a pencil. The ends look different and they have different names because they look different, okay? And so if I call this, if I say that this nucleotide, if I see that it has a phosphate, remember a nucleotide is a phosphate, a sugar, and a base. That's one nucleotide. If this phosphate is sticking out here, then I know that this is the 5' end of that nucleotide. And that's what we call that end where the phosphate sticks out. So remember, phosphate and 5 sound the same, okay? So if this nucleotide, this end is a 5' end, then that means if I just follow that down, then this G, the sugar, is going to be kind of the end thing here on that nucleotide, and that's the 3' end, okay? DNA is anti-parallel, so it's parallel, but it's anti. So if this is a strand of nucleotides and this is a strand of nucleotides and they're held together by H bonds, if that's a 5' end, then this end is going to be a 3' end. Okay, so that's what I'm kind of showing you here. If this is a 5' end, that's a 3' end. So if this is the 3' end on that DNA molecule, then this is the 5' end. Okay. Now, we have split apart the bases because we want to copy DNA and we want to make 2 molecules of DNA that is genetically identical to the original one we have here, okay? When we replicate DNA, when we split it apart, we're going to put nucleotides here in the middle, okay? So I always kind of like to draw where the new strands of nucleotides are going to be assembled. And you have to have this fact memorized. To build a new strand, you always have to build the new strand in a 5' to 3' direction, okay? This is not the new strand of nucleotides. This is called the parent strand, or you can call it the template strand. This is the original DNA molecule. You can call it the parent strand or the template strand because you use a template to figure things out. So we're going to use these bases to figure out what bases need to be added so I can make double-stranded DNA here and here, okay? New strand. These are the new strands that we're going to build. If this end is DNA is 3', when we put a nucleotide here, that'll be the 5' end, which means the nucleotide we put here, its 3' end is going to be sticking out, okay? We build the new strand 5 to 3, it's going to be made in this direction, okay? Towards the fork. This is called a replication fork. It's like a split in the road. A fork in a road is where you're on one road and it goes into 2. Okay, so this is called the fork, replication fork. This is a 5' end. When we put nucleotides together, when we put a nucleotide here, that'll be the 3' end. So that means this will be the 5' end. The new strand is built 5' to 3'. 5' to 3'. Okay? All right. Just from that information right there, you should now be able to tell me which side of the DNA that's being replicated is called the leading strand. Now you're going to put an answer in chat. Do not hit enter until I count down. So your answer is either going to be A or B. Which side is considered the leading strand? You're going to put A or B. Don't hit enter until I tell you to do so. A or B, which one's the leading? Okay. 3, 2, 1, enter. Oh, Vincent, drive me nuts. Nice, it's A, and here's the reason why you can tell that. This molecule, we're going to see here in a second, that molecule is breaking those H bonds and separating the parent strand of DNA. It's moving that away. We're going to see here that when we build 5 to 3, that means the first nucleotide is going to be put here. It's going to be complementary. The next one's going to be attached here. The next one's going to be attached here, right? See how we're building it this way? So the enzyme that's doing that is moving this way, and it's being led by helicase. So that's why this side is called the leading strand. I want you to think of the molecule that's building is being led by helicase. It's going in the same direction as helicase. So this whole side, as it's unzipped, new bases are being added right behind helicase, and we say that this side is made continuously. That's the leading strand. What do we call B?
Is it the lagging?
It's called the lagging, okay? Because it's a little more complex to make. And maybe if you think about it, like if something's really complex, it takes you a little bit longer, you lag learning it because it's so complex, okay? So we're gonna see this side, the lagging strand is gonna be made and it's gonna be a little more complex to get it assembled. All right, let's get eyes on an animation of this, okay? So I kind of taught you and highlighted some main points. Now we're going to focus on the enzymes that are going to assist in copying DNA. And I'm lagging a little bit here. Hopefully we can get this to work. All right, it looks like it's playing here. So when we're done, we should have 2 molecules of DNA from this strand, and this is called the parent strand or the template strand. Here comes the unzipper. That is a protein that is undoing the double helix. So it's called helicase. It's a protein, it's an enzyme. Let's just focus on this template strand up here. This is going to be the leading strand. And the reason why, because you see when we build a new section, we're going to build it this way towards where the helicase was going. Okay, now let's talk about this enzyme. This enzyme is called RNA primase. RNA primase has the ability to just hook on to a single strand of nucleotides, which is what this is, and start attaching nucleotides. But the thing about RNA primase, is these nucleotides that it's matching up to these nucleotides, the DNA template parent strand, those are not DNA nucleotides. They're RNA nucleotides. And RNA is different. Remember, RNA has ribose in it, and there's no thymine bases. There's U's, okay? So this primase is putting on what's called the primer. The primer is just a series of RNA nucleotides, okay? So that part's called the primer. Now, at the end of this primer, we have a nucleotide that has a 3' end. And I'm going to attempt to write here a little bit if my computer is not going to lag on me too much. Okay, so let's see if I can go back just a little bit. Notice this primer was attached. This nucleotide went on first, and then this one, and this one, and this one. So it was made in this direction. So if I asked you what this end of this nucleotide is, you should be able to tell me that this would be the 5' end. And now this is the important part, because I have a nucleotide here that is a 3' end, and this next enzyme, and it doesn't want to write, this is a 3' end. This next enzyme we're going to see can add DNA nucleotides, but it has to add DNA nucleotides to a 3' end of a nucleotide that's already on there. Okay, let me get rid of all of that and see if we can get this to go a little more smooth. Get rid of that. Okay, so here's a 3' end. This molecule is going to add DNA nucleotides. It's going to build a polymer of DNA, and that's why it's called DNA polymerase. Here's the DNA nucleotide. So I'm going to see bases that have A's and T's and C's and G's. And DNA polymerase just simply helps those H bonds form. So now we're adding DNA nucleotides. Again, we're building, look at the direction, 5' to 3' on the new strand. This enzyme will be following helicase. So it's made continuously as helicase unzips. So that's called your leading strand, okay? Now we're gonna focus on the bottom strand, the lagging strand, okay? And notice that we have a section here that's already been done, and we've got them different colors. Why don't you grab 5.3, your replication worksheet, okay? And if you don't have a paper copy, there's an electronic copy. An electronic copy is color-coded, which makes this first thing we're going to label a little bit easier to recognize, okay? If you zoom in here, I want you to find E. And I don't know how well you can tell this on your paper or how well you can tell it on my computer screen, but this little segment of nucleotides right here is a slightly different color. Can you tell that? Can you tell it's darker? Okay, because it's trying to show you that these 4 nucleotides here are different from the rest because those are RNA nucleotides. So this section right here, labeled E, is the primer. The primer has to get put on by RNA primase. And after the primer goes on, now we have a 3' end here that DNA polymerase can hook onto, and it can then add DNA nucleotides. Okay, so that's the primer. Primer got put on. Okay, and what we're going to start up here next is we're going to see here in a second that we're going to make this part in sections, and those sections are called Okazaki fragments. Here comes the next section we're going to build. Primase puts on the primer. Those are RNA nucleotides. I have a 3' end that DNA polymerase can now hook the first DNA nucleotide to. So DNA polymerase 3 is adding DNA nucleotides until it hits that next section that's already been done. We're going to see that happen again. RNA primase is putting on RNA nucleotides, and now I have a 3' end. DNA polymerase can then put a DNA nucleotide and then several of them. Okay, now we need to pause because we have a problem. Remember, DNA needs to be made of DNA, and that section is RNA. So we need another molecule, and this one is called DNA polymerase 1. Now, I'm going to use this to write because it's lagging so much. When I write 1, I'm going to use Roman numerals. And this is how I remember what DNA polymerase 1 does. I'm going to take this and I'm going to turn it into a hammer. So now I have a hammer, and hammers knock things and destroy things, right? So DNA polymerase 1 is like a hammer. It's going to come along and it's going to hammer off the RNA nucleotides and then add DNA nucleotides in that space because I need DNA to be made of DNA, not RNA. Okay, so that's kind of how I remember what 1 does. I think of that 1, turn it into a hammer and go and knock off those DNA nucleotides. Okay, and that's what we're seeing here is this enzyme is called DNA polymerase 1. It takes off the RNA nucleotides and it's going to replace them with the DNA nucleotides. So now my DNA is made of DNA. Now I'm going to hit pause here in a second because there's one important line here. You all can see my cursor, right? On my screen? Do you see that looks like there is a little gap there? Right there. Do you see that black line? Okay, so these 2 nucleotides, that's the backbone, and their backbone needs to be joined. There's another enzyme that's going to come along and it's going to glue the backbones together. And that's called ligase. Now, maybe help you remember what ligase means. Have you guys ever heard of litigation? Litigation is kind of what lawyers do. They take all these pieces of evidence and they put them together into a story as to what happened. So ligase is going to put the pieces together. It's going to seal the backbone between these segments of nucleotides so that we get a DNA molecule fully assembled. And that's what you see here. Here are the ligases. They're gluing the backbone in those areas where there was gaps. Okay. Now, let's take a look at a different image of replication. And you're going to see this one looks a lot like this one, 5.3. It also, I have another one here that's on the next page. And you can see this looks a little bit more like what's on your screen. So let's make sure we can understand what's going on here. All I want you to do is focus on the top parent strand. Okay, let's just look at the leading strand. This top part would be called the leading strand. Why? Because as we're going to see here in a second, this molecule, which is adding DNA nucleotides that are red, is moving in the same direction as helicase. Helicase is unzipping, and DNA polymerase 3 is just adding DNA nucleotides and is following along. Okay? Now, let's then pay attention to the bottom strand. Okay? The bottom strand is the lagging strand. And the lagging strand is going to be made in the opposite direction that helicase is going, but it's still being built 5' to 3'. So if I asked you to identify the end of this primer that you have right here, let's see if this is going to write, which I don't know if it's going to write. I'm not even going to try it. I'm running out of time. Is we're still building this molecule 5' to 3', but it's just away from the fork, okay? So let's I'm going to let it play here. And it's going to show a primer got put on by primase, DNA polymerase added bases. Okay, let's watch. We're going to watch this next segment, or not. Let me rewind it. A little bit more. And here we go. Let's watch the lagging strand be made in segments. On goes the primer. So that's primase putting on the RNA. DNA polymerase now has a 3' end of a nucleotide that it can attach to and start adding DNA nucleotides in a strand. Okay. DNA polymerase 1 just took off that primer, and there was a gap there in the backbone. Ligase comes and seals. All right. Couple other things that this animation isn't showing us. So let me kind of show you back with this original diagram. Okay. So we've unzipped more of the DNA. Okay, so helicase is up here. And one thing to note is that these bases, they're attracted to one another. They're both polar molecules and they form H bonds between them. So they're like little magnets. They always want to go back together. So when helicase unzips, if something doesn't hold them apart, they'll actually go right back together. So there's some other molecules that can hook on to this single strand of nucleotides on both sides, and it keeps them apart. And those are called, because this is a single strand, they're called single strand binding proteins. So you need to know their job, okay? As a matter of fact, let's take a look at worksheet 5.3. And can you tell me which letter do you think is representing single strand binding proteins?
B.
There you go. B would be the single strand binding proteins if you want to label those. Okay. Now, another enzyme they like to talk about is, I'm going to bring this original one back here. There's another enzyme that is going to be located more up here ahead of helicase. It's at the top. The great thing to remember about top is that's the first 3 letters of this enzyme, top. It's topoisomerase is how you pronounce it, topoisomerase. And the job of topoisomerase is to keep DNA from breaking. And here's the reason why DNA can break. If you unzip a twisted molecule, it makes tension on the molecule upstream. Let me show you that. And that's the reason why I've had this taped. Can you guys see that I have a double helix here? Okay. Now watch what's going to happen to this DNA molecule. So if I unzip it, so we unzip it, we made a replication fork here and here. Okay, single strand binding proteins would be holding it open, and helicase would be this. Okay, so I need about 5 different hands to do this. So helicase is unzipping, and watch what's going to happen as it unzips. There becomes a point where I can't put enough pressure to make this go anymore. Is because, do you see how tight my string got? You see all that tension there? So I'm going to unloosen the side. When helicase unzips, tension develops upstream. And tension, watch what happens. You see the coiling? Those are called supercoils because the DNA has so much tension on it, okay? And so there's an enzyme that assists with relieving the tension, and that's the topoisomerase. Like I said, I need about 5 different hands here. Topoisomerase is like these scissors. It has the ability to snip DNA upstream, unravel it, and reattach it so we don't have as much tension up here. because if this tension isn't dealt with, the DNA is just going to break. Okay, so that's the job of topoisomerase. Now, with that in mind, what we've covered so far, instead of sending you guys back to breakout rooms, what I want to do is we're just going to all work together on 5.3. And I'm not going to tell you any answers. You guys are going to figure them out. And you know what? Let's make this interesting. You personally can only give 2 answers to the group. So you better share the wealth. Okay? So here we go. Let's try to figure out some things. First of all, let's figure out which is the leading strand and which is the lagging strand. So your options are either K or J. Which one do you think is the leading strand?
The leading strand is K.
All right, Vincent, there's your one. And the reason why is because you can tell that this arrow means that we're making double-stranded DNA in this direction, and helicase would be unzipping in that direction. So K is the leading strand because helicase is leading the way. Okay? Which means J automatically is going to be what?
Lagging strand.
Awesome. Who said that one?
Raina.
Thank you, Raina. There's your lagging strand. So you can label your J. Okay? A is on both sides. So what is the name of the enzyme that is literally just putting and making the DNA using DNA nucleotides?
DNA polymerase.
Right, because it's building a DNA polymer. So A's are going to be the DNA polymerase 3. You don't have to know 3 and 1 for a test. If you just say A is one type of DNA polymerase, I'm good with that. But I think students actually remember and learn it better if you know that there's 2 different types of DNA polymerases. There's a 3 and there's a 1. They have different jobs. Okay? All right. So we already talked about E, and we said E was the primer, right? Okay. Let me give you a hint here. D is the one that they're trying to show D is putting on the primer. So what's the name of that enzyme?
Primase.
I think you're done, Vincent.
Oh.
Don't make me mute you. Yes. So D would be primase, which is a protein doing the job. It puts on that primer, and that primer is made of mRNA. Okay. This one, F, is trying to show you that here at the beginning of this segment, there was a primer and it's been removed. And DNA is going to be filled in the spot. So what's the name of the enzyme that can remove the primer and add DNA nucleotides?
Is it the other polymerase?
That's the hammer, DNA polymerase 1. Take the 1 and turn it into a hammer. Okay. So if this was, as Raina said, this was the lagging strand, then this little section that was kind of made at one time, that's what L is saying. What do I call a section that's been made on the lagging strand? And there'll be another one here and another one here.
Is it the fragment?
And they're called Okazaki fragments. And it's named after the man that determined that one side of the DNA was replicated in pieces or in segments. So he named them Okazaki fragments. Remember, if you discover it, you get to name it. So I can't wait, for an enzyme that I'm going to teach about in the future that's been newly discovered, and it's going to be named after one of you brilliant people that did it. Okay. All right. G, if you look very closely, there's a hole there in the background. There's a gap. So what do we call this enzyme that seals gaps in backbone?
Ligase.
Very good. There's your ligase. Okay. Just to check a little bit for understanding, put an X where I would find topoisomerase. Put an X where you think I would find topoisomerase doing its job. Topo, remember it starts with top. It's going to be the very, very first thing. So topoisomerase is going to be working up here cutting apart nucleotides, unraveling the DNA, taking the tension off, or it's going to break. We don't want DNA to break. Okay, it looks like we need to figure out the ends. And again, this goes back to the most important rule, because if you don't remember this, then you're going to get your lead-in and your lagging mixed up, is that when we put on a new strand of nucleotides, so this is a new strand, and then we're putting on a new strand here, you always build the new strand in a 5' to 3' direction, okay? So if the arrow's pointing this way, which means that we're replicating in this direction, the first one that went on would be the 5' end. So anytime we end, that would be a 3' end, okay? So watch my pencil here, because if this is a 5' end, let's keep following. So I would be what?
Three.
3, which means H has to be the 5' end. Okay? All right. How about we keep going? 2, I'm going to let you do on your own. Explain why one strand of DNA can be replicated continuously, while the other must be made in fragments. And I will give you one little hint. That's kind of the most important thing that you have to remember. And it's because of this, one's going to be made towards the fork and the other one will be made away. Okay. All right, let's use this information to kind of answer another question here on the last page. So hopefully this makes sense to you, that we had a DNA polymerase, it attached here, we're building 5 to 3, it added a C and then an A and a C, okay, and it worked this way. So now I have double-stranded DNA. This one will go to one daughter cell, thanks to mitosis, when it lines up those chromosomes that have been copied. And then this side, this DNA molecule will go into the other daughter cell. So I have 2 daughter cells that are genetically identical with the same DNA that was in the original cell, okay? Now, again, if I'm replicating away from the fork, this is my lagging strand. Do you know why I have U's here?
Because it is a RNA.
Right. So when RNA primase hooks here to the single strand that's been separated, it can only add RNA nucleotides. And my pencil can't write T if I'm writing RNA. A goes with U. A goes with U, okay? But now I have a 3' end where DNA polymerase can hook onto and now can add the DNA nucleotides, okay? All right. Again, that primer is going to be taken off, though, by DNA polymerase 1 and replaced. So now it's not going to be U anymore because A goes with, it's going to put thymine as the base, or the nucleotide will have thymine. Okay? Hopefully that makes sense. Now, last thing that I want to kind of point here is, remember, these are my new strands of nucleotides added to the parent strands. Okay? Notice my color code. This is important. Is the original DNA molecule, has it been completely disassembled and it's gone? Do you see that the original, so here I have a new molecule of DNA, and it's really just made of the original. So note This model is called semi-conservative. Conservative means to keep. Semi means kind of, like semi-sweet chocolate. It's kind of chocolate, kind of bitter, okay? So it's called semi-conservative because when we're done replicating our 2 molecules of DNA, the original is kind of still there and intact. This half of it was just the original DNA that was in that parent cell. Okay, so it's semi-conservative replication. All right, so let's take a look at, let's go to number 8. This is on the 3rd page here. So it says this diagram shows one of the 2 DNA molecules produced through replication. So it's just showing you like this. This is a double-stranded DNA. It's just showing you one side. Okay, so here's one DNA molecule. It says, was the strand replicated as a leading or a lagging strand? Do you see fragments? Do you see gaps? Lots of them? It's because that strand was made in pieces. So this part was made first, and now it's done, and then this part got added, and then this part got added, and then this part got added. We've got all these holes here because we're making it in pieces. So would that be leading, or would this be a leading or a lagging strand that we're looking at here?
It would be a lagging strand.
Very good. Okay. If I ask you this, which side of the DNA was the parent or the template strand? This top strand of nucleotides was the parent strand, or the original DNA molecule, or the bottom?
The bottom.
Mm-hmm. That's the original molecule. There's nothing to fix on it. That's the original. We just separated the original. Based off of kind of what I just did here in this little simple paper model in that section right there, have the primers been removed?
Yeah, because you can't, there's no RNA
Right.
left.
If the RNA was still on there, we would see some T's would have been used. So we can probably deduce that all the primers have been removed and replaced. And really, we're waiting for the last step. We're waiting for the ligase to come and take these 2 nucleotides and join their phosphates and sugars with a phosphodiester bond. So we need this to come in and happen, right? So D says, what enzyme is needed to finish the process of replication on the strand shown? Okay. And then let me ask you this. This is one DNA molecule of the 2. What's the fate? Where are these going to go? That's F.
Isn't it that when you have replication, one of them is going to go to one of the cell and the other was going to go to the other cell?
Mm-hmm. Yep, that's the fate. And that's the whole reason why this would ever happen in a cell, because the cell's in S phase and it needs to copy its DNA, make 2 sets. All right, whew, finally got through that one, even though my computer was lagging like crazy. So, all right, guys, you know where to find me in the afternoon if you need anything, okay? Be studying and getting caught up. I did post the 4th and final lecture, which the good news is, biomed students, this is good news for you. This is the first time you're gonna know more than the pre-AP students because it's over PCR. There's no PCR, right? All right, cool. All right, we'll see you guys tomorrow. Have a good afternoon.